Okay guys, this is a video about ATP. Um, this goes along with chapter 8 in your book. It's not terribly long, so hopefully we can make sense out of this. I think you'll think this material is pretty simple. Alright, so it's all about ATP. Alright, sorry about the sound effects. I hope they're not recording super loud. But the point is to make ATP. Right? So remember that we're talking about mitochondria breaking down nutrients in order to make ATP and the cell needing to use ATP. So we're going to talk about what makes ATP so usable as an energy source. Okay, the energy needs of life. Of course, life requires a lot of energy for a lot of different things. Organisms are endergonic systems, which is to say if you don't eat, you die because you run out of energy. So endergonic and exergonic definitions are on page 146. So endergonic means is a reaction that absorbs free energy from its surroundings. For example, in photosynthesis, plants are storing energy, and that's an endergonic reaction. So, what do we need energy for? Synthesis, building biomolecules that are necessary for the structures of living beings, for reproduction, for movement, for active transport across cell membranes, getting stuff across that cell membrane in the processes that require energy, and temperature regulation. So that's the point of having energy, is to do all of these things. Right. Where do we get energy from? Okay, the work of life is done by energy coupling. And that's to say that we're combining these exergonic and endergonic reactions. So I'm going to explain this in one sec here. All right, so we're going to use exergonic catabolic reactions to fuel endergonic or anabolic reactions. So this picture here is showing that coupling. So if this first part is digestion, so we're taking some large bunch of molecules, okay, and then breaking them down into their parts. So we're taking polymers and breaking them into monomers, and the energy that comes from breaking those bonds. Okay, then, all right, the opposite of that is to take monomers, use that energy and create a polymer. So these are opposite reactions. In one, we're having energy outward, and in one, we're using energy. So one is catabolic and one is anabolic. So why don't you pause for a second right here before I tell you the answer and tell me which one, digestion or synthesis, is exergonic and which one is endergonic. So pause right here and see if you can do that. Okay, so in the first one, it's exergonic because energy is released outward. The EX, E means outward, okay, END means inward. And in synthesis, it's endergonic because we are using energy to synthesize something. Okay, so digestion is catabolic and synthesis is anabolic. Okay, fueling the body's economy. And here's how we put together the things we've been learning about cells and what we've learned about the chemistry of life. Right? We eat high energy organic molecules. So our food is carbohydrates, lipids, proteins, and nucleic acids. We take all that in the food that we eat and then break them down. So digestion is catabolism. So digestion is a catabolic reaction. So we're going to capture released energy in a form the cell can use. Right? And we need an energy currency, a way to pass the energy around. So often ATP is referred to as the currency of the cell, like money. It's a way to spread this around in a closed system. So it's a short-term energy storage molecule. And the penguin's going to say, oh, the penguin comes next. Hot stuff, all right? So we're going to eat food that's more complex, and we're going to break it down. All right, and when we break it down, we get energy from it, and then we're going to use that energy to do other life processes, as well as synthesizing the molecules that we need to make up our cells, for reproduction, for movement, for everything else that we do. Okay, so ATP, of course, is adenosine triphosphate, and it's a nucleotide. Right? A nucleotide, remember, is a sugar, a phosphate, and a nitrogen base. So in this case, you can see we have, okay, adenine and ribose and a phosphate. Now if we just have as a building block here just the one phosphate, then we have adenosine monophosphate. Okay, and then we're going to build on. Okay, we're going to add on another phosphate and have adenosine diphosphate. Then adding one more phosphate group. Okay, we go from ADP to add a phosphate group and end up with adenosine triphosphate. So here's the completed molecule. And I want you to notice here that these oxygens have a negative charge, because that's going to become important in a minute. And when we add phosphates on in this way, so remember we started with just the one, and then we added another and another. When we do that, it's an endergonic reaction. Okay, so energy is being stored in these bonds for later. So just what's important here is that you do need to know the parts of adenosine triphosphate, that it's made of adenine, which you know is in RNA and DNA, and it has a ribose, the 5-carbon sugar, and then 3 phosphate groups once it's ATP. But it starts out as AMP, then becomes ADP, and then becomes ATP. And that reaction of going from being a monophosphate to a triphosphate is endergonic. All right, so how does ATP store the energy? This is where some of those charges are going to come into play. So you've got this AMP sitting here. Right, it's got the negatively charged phosphate group here. And do you think it's easy or difficult to add another phosphate group on there? What do you think? So if this is negatively charged and I'm going to take another negatively charged phosphate group and attach it there, you think that's hard to do or easy to do? Well, I'll tell you, it's not easy to do. It's difficult. It takes a bunch of energy to add another phosphate group because the same charges are going to repel each other. So we're going to become more or less stable. What do you think? We're going to become less stable. It's going to be more unstable. So because the next phosphate is negatively charged, it takes energy to add it, and it's going to make it more unstable. So we're going to add it here. Okay. Boing. All right. So now we're at ADP, adenosine diphosphate. So we still have these two negatively charged phosphate groups. It's highly unstable. So do you think it'll be easy or difficult to add another phosphate group to that? It's going to be super difficult to add another negatively charged repelling, and it's going to make it even more unstable. Kabang! All right, so that makes it a super unstable hot stuff kind of molecule. It wants to give off that phosphate as soon as it can. It's not going to stick around. It doesn't want to store it up. It wants to give this phosphate away at the soonest opportunity. All right, so each time we add a negative phosphate, the more difficult it is to add. So a lot of energy is stored in each of the bonds of these phosphates. And the highest energy will be in that third phosphate that we've added. So this last bond is the one that's got all the energy that we want. The third phosphate is the hardest group to keep bonded to molecule. It wants to give it up. And the bonding of these negative phosphate groups is unstable. It's spring-loaded. Okay, ready to bolt. So that's how ATP stores energy. It's stored in these very unstable, hot, ready-to-go phosphate bonds. And ATP really wants to give up that last phosphate. So the last phosphate's going to pop off easily and release energy. And here comes the penguin. I think he's a bit unstable, don't you? So the instability of its phosphate bonds makes ATP an excellent energy donor. All right, so that's how ATP stores energy. The next step is how does ATP transfer energy? So when we're going to transfer energy, we're going to want to ditch this third phosphate group. We're going to be going from ATP to ADP. So we're going to ditch this phosphate by phosphorylating. So you can think of the third phosphate as the bad boyfriend that ATP tries to dump off on somebody else, and that's phosphorylating. So the other molecule would be considered phosphorylated because it's ended up with that phosphate group that ATP is trying to ditch. All right, so these oxygens that are on the phosphorus are super electronegative. So when you have a lot of oxygen, you have a lot of electronegativity. So we're going to ditch that phosphate. Right? And energy gets released. Well, how much? Okay, 7.3 kilocalories per mole. Okay, so in your book, they show you that we use this triangle in G to mean the change in free energy is negative 7.3 kilocalories per mole because it's given off. And that energy can be used to fuel other reactions. And again, this is phosphorylation because this phosphate isn't just going off into space, it's being dumped onto another molecule. So the released phosphate can transfer to another molecule. And it will destabilize the other molecule. And you probably remember this kinase term because it came up when we talked about cell signaling. The enzyme that phosphorylates is usually called a kinase. So let's go through an example. Oh, well, just to summarize this again, how does ATP transfer energy? Is that ATP wants to ditch that third phosphate. It's like a boyfriend the ATP wants to drop off on somebody else. So off goes the phosphate and ditches it onto somebody else, and when it does that, 7.3 kilocalories per mole are released. And that is called phosphorylation. And this is a hydrolysis reaction. Okay, so water is required for this. And if you want to see that piece of this reaction, it's on page 148 in your book. Page 148 shows you the water requirement for the hydrolysis aspect of this reaction. Okay. Eventually, the phosphate is going to get dumped by the other molecule too, and then will go alone into the cytoplasm again. All right, because then remember that ADP is eventually going to pick up a phosphate. Okay, and we'll go through that again in a second. So, all right, an example of phosphorylation, building polymers from monomers. All right, how does this happen? All right, we've talked about this a bunch. All right, so here we have two monomers, and we're going to synthesize them into a polymer, right? And we remember that water is a part of this reaction, but the water doesn't just come off on its own from this, okay? It gets pulled off by phosphorylating the monomers. Okay, I'm gonna end part 1 right here and pick up right here on the next one. These videos are a little easier to work with when they're a little bit smaller. So this is the end of part 1, and we'll pick up right here in part 2.